Nucleic acid. Last class we discussed about the nucleic acid and the structure and the function of DNA and RNA and the biological importance of the nucleotides. So I am assistant professor of the Calcutta Institute of Pharmaceutical Technology and Allied Health Sciences. First of all, we convey the message all the listeners must be stay at your home at this time to prevent and save for your life against the coronavirus. Today we start with the nucleic acid and basic structure of the DNA. The discoveries we are discussed in the first class. The Friedrich in the 68 night, which is discovered the nucleic acid component from the nuclear material of the salmon, sperm, and the name is the nuclein, and which is called the nucleic acid. And John 1920 it proved that there is a two type of nucleic acid, one the deoxyribonucleic acid joined in DNA and ribonucleic acid in the RNA. But 1935, very recent, last 85 years ago, J.D. Watson and F.H.C. Crick, on the basis of the information available, not only the proposed in the double helical structure of the DNA, but also suggest the Crick terms central dogma of molecular genetics, and which is the state that the genetic information flows from DNA to RNA and RNA to a protein. This is our life process or central dogma of life. This is the structure of as per the design by the Watson and Crick. 1953 he got the Nobel Prize also, just like a helical structure. And this is the central dogma of life we discussed in the previous classes. Rosalind and Franklin, which is studied the structure of DNA by the crystal using the X-ray, and found the crystal contain the regularly repeating subunit. And the X pattern produced of DNA suggests that the DNA contains the structure with the dimension of 2 nanometer or 0.35 nanometer, 3 angstrom or 3.4 nanometer. And this dark structure at the top and bottom inside the some structure was repeated and suggesting the helix. The Franklin and Watson and Crickson, J.D. Watson and Crickson in the 1953 using the Chargaff base and the Franklin X-ray diffraction data to construct the model of DNA. The model shows the DNA is the double helix with the sugar phosphate backbone at the outside, and it is prepared by the nuclear base of the inosine, and it the structure that fit and the spacing estimated from the X-ray diffraction data. And the Chargaff rules AT and GC, 50% of purine and 50% of GC, with a complementary base pairing rule, and the prepared base can occur in the others and given to the other switch. Chargaff rule the base pairing AT with GC, thymine is replaced by uracil in case of RNA, and base are bonded each others by the hydrogen bond, and discovered because of the relative percentage of the bases. Notice AT is similar to GC similar. History of DNA, the Sir Ochoka with the 1903 Nobel Prize in the medicine after the discovery of RNA is synthesized. And the Carl realized that RNA can catalyze the 1967 and proposed that the earliest form of the life relate on the RNA both to carry genetic information and catalyze the biochemical reaction of RNA world. In the nitrogenous, the silent endogenous genes and plant known as the result RNA interface, and the same year they discovered the gene regulatory RNAs lead to the attempt to the develop of the drugs made of the RNA, and like siRNA, small nuclear RNA in the silent gene. The nucleic acid, two type DNA and RNA, the building blockers of the nucleic acid or nucleotides. Nucleotides, monomer of the nucleotides, which we discuss about the monomer, the base, sugar and phosphate. Basically N base, that means nitrogenous base, that's why it is called the N base, phosphate, phosphoric acid, and sugar ribose or deoxyribose in case of. What is DNA? And DNA is the molecule which is the component of the genetic instruction used for the development and functioning of all known given to the organisms and many viruses. This is the basic structure of the DNA. First, it contains, it's a double helical structure, right-handed double helical structure. Bonded one strand is composed by the another strand joining by the hydrogen bond C with triple bond and AT with double bond, and having it two groups. One is the minor groups and it is the major groups. One is the minor left hand side 1 nanometer, it is the minor groups, and 3.4 it is the major groups per helix. It is a 3.1 major groups and minor groups, and the diameter between the DNA molecule is the around 2 nanometer or 3 point, sometimes some books also 34 angstrom also. So basically major groups which 3.4 nanometer, minor groups 1 nanometer, and the diameter 2 nanometer, and it is composed by the 5 prime to 3 prime. Basically you can see it, this is very important, it is an anti-parallel. 5 prime to 3 prime or 3 prime to 5 prime always. If it is right 3 prime first strand, 3 prime to 5 prime, the second strand will be contain 5 prime to 3 prime. It will be seen, but it will be very carefully it is seen it the 5 prime to 3 prime. That is why the DNA is the anti-parallel. Nucleoside structure: phosphate, sugar, ribose, deoxyribose, and bases purine, adenine, guanine, pyrimidine, ultimately nucleotides. The ribose sugar which is present 5 prime, actually the difference between the same, the deoxyribose, the two position it will be thus about devoid of oxygen in case of deoxyribose. Purine bases adenine, thymine, adenine to form a thymine, and cytosine with guanine with the guanine. DNA structure: deoxyribose specific, adenine, guanine, and cytosine, thymine. The base pair ruled by the Chargaff and the form a double helical structure. RNA sugar, ribose sugar, thymine, it replaced by the uracil and single strand. Nucleus DNA, RNA, what is the structural shape? That is the difference between the RNA, double helix or alpha helix, linear or single strand. Strand number 2 in case of 1, sugar, deoxyribose, here ribose, N base, ATGC, here AUGC. It is anti-parallel, it is not anti-parallel. DNA used for the genetic information. Actually all the information is stored in the DNA, and DNA produce different types of RNA to protein. That's why DNA is called the reserve bank of genetic information, not RNA. Be careful, remember it. DNA is the reserve bank of genetic information. All the genetic information, all the structure, all this body, every parts, every function, every enzymatic action, every protein action, every hormonal action can be controlled by the DNA. That's why even their shape, size, body, character, everything, but some character will be issued or gain in the environmental factor, but other also, but hereditary, all these information coming from the DNA. That's why it is called again the most genetic material content in the DNA, and the DNA is the very important, so it is the stored in the nucleus. It is never leaves the nucleus. The DNA gets converted to RNA in order to move and into the cytoplasm. The cytoplasm meets up to the ribosome. There already they synthesize the proteins and store the genetic information, maintain the growth, repair, everything. Your body growth, that will be constructed. Apart from you can see the growth, that is not done by the DNA, but all these functions of the body can be instructed by the DNA. And control of the cellular activity, contain the protein coding, ensure the daughter cell to gametic receives the exact genetic information is formed by the DNA. And the abbreviation deoxyribose, it is the polymer of deoxyribonucleic acid, and it is a thread-like structure and combination of the large number of nucleotides and joined together. And this polynucleotide contain the genetic information and gives us the chemical and physical property of the organisms. And what is the location and isolation? Basically, it is located in the chromosome, found in the chromosomes and mitochondria, chloroplast of the cell, and it is present in the every living organism because it contains the genetic material. Isolation from the virus, bacteria, thymus, glands, spleen, blood, hair, skin, etc. Size of the DNA, generally the great variation. Actually, this DNA all the organisms like organism prokaryotic to eukaryotics, and in case of even the human beings, the basic composition are the same. All are contain ATGC base pair nucleotide, ATGC ribose sugar. But in case of cow, goat, and all the animals, what is the difference between the animal DNA and the human DNA or other DNA? Basically, all components are the same, but difference. Be careful. Only the sequence is the difference. That's why the tiger is tiger, and human is the human. Human DNA sequence is different from the tigers or goat and cow. If possible, the tiger DNA is incorporated human, the human behaves like a tiger's, or the human DNA, if possible, human DNA incorporated into the tigers, and the tigers can behave like a humans. So that is the things, but it is not possible because their DNA sequence, only the sequence component is the same, but the sequence is the difference. I think you are understand. And only the 1 micrometer long in the simple structure of similar virus with 5 to 6 gene, and it can also extend 2 meter in human DNA. Be remembered, 2 meter, our human DNA length is the 2 meter long. 2 meter, whereas the virus 1.7 micrometer. But how to incorporate 2 meter long substances? How to incorporate it in the nucleus of the cell, not in the body? It is a part of the party. The small molecular cell, it is not seen in the naked eye. It is in the microscopic structure. After this, it is nucleus. Actually, it will be possible to 2 meter long DNA which incorporated in the nucleus, only possible because it's the helical structure. Or spiral, if a two strand are helix supercoiled or right-handed supercoil, this structure, the helical structure of these two, that's why their chain length will be reduced on twist between the two linear strand. And the size of the human DNA inside the chromosome is the 200 nanometer. And the structure of the DNA, all of you know it, the sugar pentose, the three-dimensional structure, thymine, guanine, cytosine. And the DNA is the genetic material for the circumstantial and the evidence, and freeze the mutagens of the effect of the UV light and peaks up to 53.7 nanometer, the peaks of the absorption UV light, which is discussed in the practical class also. UV light and UV money, that means ultraviolet light, their highest absorption is the 253 nanometer. 250 nanometer. Ribosome, the pentose sugar, spot the difference between the ribose sugar, same bonding. The sugar, the base sugar phosphate bond. Actually, all nucleotides are esterified with the phosphodiester linkage, adding with the base first carbon, and they are important to determine the genetic information because of fossil also. This is the complete structure of the DNA. Their base is composed by the hydrogen bond, in case of GC or CG by 3 double bond, 3 hydrogen bond, and in case of AT 2 double bond, and all the nucleotides component which is joined by the PP or phosphodiester bond, and it is a very high bonding. Even the fossil 1000 after 1000 years, this phosphodiester linkage never cannot be break out, and that is the identification of which fossil and which group of fossil or which group of animals by the phosphodiester linkage. And this is the twist Chargaff rules. Just you see it, you can understand it, you carefully see it, which is in case of man, sheep, turtle, and the animal, plant, and bacteria all are G and C. They are base pairing, mole parents, and base ratio and symmetry ratio, all this given in the chart. DNA is the made up of 2 strands of polynucleotides, and the sister strands of DNA molecule is run opposite direction or anti-parallel. They are joined together by the base, and each always pair with the T, and G pair with the C. It is called complementary base pairing role and hydrogen bond. Type of DNA, 2 types of circular, non-circular, and circular DNA. In case of eukaryotics, there are end of DNA at the cohesive, so it is joined to the circular DNA, the mitochondrial DNA, chloroplast and prokaryote mostly in the form of plasmid, whose replication do not depend upon the genetic information of bacteria. This is called the circular DNA in case of prokaryotics, the DNA which does not have a few ends, but in case of eukaryotic it is a linear, and this DNA present in the nucleotide region of the prokaryotic cell. Non-circular, there is two anti-parallel strand DNA twist around each other and to form a helical structure of the double helix. The basic difference there is in case of DNA is be classified into three groups. We remember it. First of all, the A form, B form and Z form. A form, B form and Z means actually zigzag. And what are the difference? This is very important in case of examination. Write a short note on A form or B form and Z form. And in physiologically, B form is the most common form of this. What are the difference characteristics of these three types of DNA? First, helical sense. Right-handed and left-handed. Repetition of unit, 1 base pair, 2 base pair. Rational base pair 32.7 degree, 34.3 degree and 60 degree. Means base pair turn 11, 10.5, 12. Indication of base pair in axis 19 degree, 1.0 degree, 9 degree. Glycosyl angle anti-antipiral. Nucleoside phosphate and phosphate distance 5.9, 7.0 average and 5.7. Sugar precursor 3.32 and helical diameter 23 angstrom, 20 angstrom and 18 angstrom. These are the basic difference between the three form of DNA, A form, B form and Z form. Suppose a short note on A form, you can write only the characteristics of A form. B form, which is write only the B form, and Z form, which is only write the Z form. And sometimes difference, you can difference. This chart will be enough for the answer. This is the structural difference: A form, B form, and physiologically active out of three form, and C form is the circular and Z form. And physiology, another most important factor, which is the physiologically active. B form is the most physiological form of the DNA which is act on the body. So I think you can understand what is the nucleic acid, what is the structure of the nucleic acid, both DNA as well as the RNA. I think all of you understand. If you not understand any question, please click on my comment box. And thank you all the listeners for your patience listening this video. Thank you again all my listeners. If you understood, please subscribe my channel.